Thanks so much, Dr. Hufnagel, for that wonderful introduction. And thank you all for coming today to hear me talk about this topic, which I hope will be interesting and informative for you. So I'm going to start with my disclosures. And then my goals for today is hopefully that you will learn about some new immunotherapeutic approaches to cancer treatment. And I hope that I will explain them well enough to you that you will understand how they work, and also really understand what the urgent questions are in cancer immunotherapy, and so that we in laboratory medicine can help to address the urgent needs that are present. So here's the outline for today's talk. I'm going to spend quite a bit of time on just general cancer immunotherapy and some relevant immunology background. I'll tell you about the Cancer Immunotherapy Trials Network, which we've been a part of, IL-15, which is a novel agent that's been used in a number of phase 1 clinical trials, and that finish with a biomarker, which you may or may not have heard about, myeloid-derived suppressor cells. So we'll start at the beginning. The goal of cancer immunotherapy is to expand and or activate the patient's adaptive immune response to specifically target or kill cancer cells. And this can be done through a number of means, modalities, and really today I'll just focus on two, and that's with antibody therapy as well as T-cell growth factors. So in December of 2013, Science magazine proclaimed cancer immunotherapy as the breakthrough of the year. And so why did they do that? Well, at that time in 2013, there have been some remarkable results that have been reported in cancer patients using multiple immunotherapies. And I think part of the reason was that there were multiple modalities being used, and these were used in the treatment of patients with very advanced cancers, and they were seeing remarkable results. So the first area was for chimeric antigen receptor therapy for B-cell leukemia, and I can tell you that I'm not going to talk about that at all today. We've heard some very nice talks on this at this venue previously, but I'm going to focus on the anti-CTLA-4 and anti-PD-1 therapies that at that time had been tested and approved for advanced melanoma. And these are the checkpoint inhibitors that I'm referring to that was in my title. So in case you think that cancer ideas about how to treat cancer using immunotherapy are relatively new, I'm going to take you back in history to 1891 to William Coley, who was a bone cancer surgeon at the New York Cancer Hospital. And he was affected greatly by a case that he was unable to treat successfully. It was a young girl with soft tissue sarcoma. And after treating this patient, he decided that he was going to try a different approach. And so what he did was he decided to focus on this observation that had been made since the 1700s, actually, that patients with cancer who had concurrently had a superficial strep infection of the skin had evidence of tumor regression. So it took a few years, but he put together a mixture of heat-killed strep pyogenes and Serratia marsensins and injected them into tumors daily for weeks to months. And he also got a lot of his colleagues to help him to do this too. And interestingly, now in retrospect, we can look back and we read about what the side effects of this treatment were, and they reported high fevers and chills within the first 24 hours of starting these injections. And now with the retrospectoscope, we can say that this was probably the result of cytokine storm. And here, in this next part of the slide, I can show you what the results were that were tabulated by others after his death, although he did report the results of his treatments while he was alive. So interestingly, the most amenable to therapy was the soft tissue sarcoma. They did test lots of other patients with different types of cancers, and they weren't as responsive as the sarcoma patients. But if you put them all together, that out of the 104 patients, 16% were disease-free but lost to follow-up after 5 to 10 years. And 14% were disease-free but lost to follow-up at 10 to 20 years. And 21% were disease-free for up to 20 years. And this is pretty remarkable for Depression era treatment. So let's put this in the context of the evolution of cancer therapy in general. And you can see it kind of starts in the mid-1800s with the availability of anesthesia that allowed for major surgeries to remove tumors. And then there was Coley's toxins, which we just talked about. Shortly after that, radiation became available to treat patients with cancer. And then that was slowly followed by hormonal therapy and chemotherapies. So why did this stop? Why didn't people keep doing this? And I don't think anybody really knows the answer to that, but I am hypothesizing that it was because people didn't really understand how this worked. There wasn't the immunological knowledge and framework to explain this. Plus, it was limited to this subset of patients who had soft tissue sarcoma. So we then went along for a number of decades now using the combination of surgery, radiation, hormonal therapy, and primarily chemotherapy until about 1997, when the first antibody therapy was FDA approved, and that was rituximab. And then in 2001, we had the approval of imatinib, which was a small molecule for targeted therapy. And then finally in 2011, the approval of anti-CTLA-4, which is what we're going to talk a lot about for the rest of this talk. So previously, these anti-cancer drugs indiscriminately killed proliferating cells, but now we have this big paradigm shift where these new agents are enhancing anti-tumor immunity. And interestingly, the more we study this, we find that some standard anti-cancer agents also have immunomodulatory properties. So I want to take a moment now to just review a little bit about immune responses. We can think about them in two sort of major categories: the innate immune response and the adaptive immune response. And this is just a review for most of you. But I like to think about the innate immune response as the first responders. The first responders are very quick, and they recognize shared structures. Diversity is pretty limited, and memory is really restricted only to the NK cells, which are categorized as an part of innate immunity. But the innate immunity sets up the adaptive immune response, which I think most people would think of as a little bit maybe more refined. It takes a little bit longer to get there, but you have antigen specificity, and it's exquisitely specific. You have incredible diversity of response, and you have memory. And so these are characteristics of a system that would be ideal for treating something like cancer. But there are some biological challenges, and they include, are not limited to, but include cancer cell-induced immune suppression. Cancer cells are very good at avoiding the immune response and protecting themselves from being eliminated. There's the issue of immune incompetence. As we all know, most cancer patients are older, and as we age, our immune systems become less competent. And then most of them are subjected to standard chemotherapy, which is oftentimes lympholytic. And then there's the thorny issue of immune tolerance. So, as most of you know, immune tolerance is a central paradigm of immunology. And so the adaptive immune response, the T cells and the B cells that develop in all of us, are subjected to this selection process. And the first process is termed central tolerance. And the T and the B cells, respectively, are developing these organs, the thymus and the bone marrow. And it's there that these cells get to test out their new T cell receptors and B cell receptors. And if it turns out that they're overly reactive to self-antigens in those areas, then they get deleted. So we're getting rid of the really bad, very reactive cells by central tolerance. But the immune system has to balance, the ability to have this really diverse repertoire to recognize any potential foreign antigen with a little bit of autoreactivity. And so the immune system has set up a backup plan where there's peripheral tolerance and control of inappropriate reactivity in the periphery once the B cells and the T cells leave the bone marrow and the thymus and go out in the periphery to survey all of our tissues. So there are multiple mechanisms of active suppression that we'll talk a little bit about too. But the thing that I want to remind you of is that tumors are really essentially self. So that makes things hard. And when we think about tumor antigens, they're mostly overexpressed, non-mutated self. Fortunately, there are some situations where there's mutated self, and that makes things a little bit easier for the immune system to recognize. There's some antigens that are organ-specific, others that are just expressed in fetal life and then get re-expressed in tumors, some that are common among different tumors. But the important thing is that through the work of many investigators, we now know that there's pre-existing cellular immunity to these tumor antigens. They're present, but usually they're inactivated. So now I'm going to take a moment to show you one of my favorite sort of schematics. And so really, the reason I like it, it shows you the kinetics of an immune response over time. So time is on the x-axis, and cell or pathogen density is on the y-axis. And what you can see at the very left bottom is the beginning of the infection, primary infection by, in this case, a viral pathogen. And as the infection grows and the pathogen replicates, here, you actually don't see it here, but there is an induction of innate immune response. And this is followed by the induction of an adaptive immune response. Sorry, the cursor doesn't work very well. So that's shown, the adaptive immune response is shown in gray. And so you can see that as the pathogen replicates, so too is there a response from the cells that they've recognized and become activated and are proliferating in response. And once the pathogen, and also during that time, I don't want to forget that there is differentiation of the responding cells so that they are acquiring effector functions. And so using these effector functions, then the T cells are able to limit the growth of the pathogen, and eventually, through various mechanisms we're not going to go into, the pathogen is eliminated. And by that point, then, the immune system realizes, well, I don't really need an army anymore, and so I'm going to contract this population of responsive cells. And what happens with time is that the very best responders become memory T cells, and they survive for years in the host until there's a re-challenge of the same pathogen, in which case then there's a pretty quick response that then aborts this reinfection pretty quickly. So that's a very long explanation for what I consider to be the effective, the important steps in generating an effective and durable T cell response that are relevant for this discussion, and that is that there's this very important initial step of recognition of an antigen, activation of the T cells, proliferation of those T cells to build that army, the acquisition of effector function, which includes things like secretion of cytokines, direct killing of infected cells, and it might be direct killing of tumor cells in the case of tumors, and then provision of B cell help, because we all know that antibodies are important too, and then finally the generation of memory. So we're going to back up and really go into detail about that first step. So starting the CAR, which is and I'm referring to the T cell here, which is shown on the right. The tumor cell, or the antigen-presenting cell, is shown on the left. And the main interaction I want you to focus on at the moment is right at the center. And the T cell receptor is shown here in yellow, and that's recognizing the MHC class I molecule containing a peptide from the antigen of interest in its binding groove. And you can see that this interaction is very snug, and that leads to a signal to the inside of the T cell that should lead to T cell activation, except that in the absence of any other signals, it doesn't. So signal one enough by itself is not enough by itself. And so there is actually a second signal that happens, which is the interaction of CD28 on the surface of the T cell with CD80 or CD86 on the surface of a tumor or antigen-presenting cell. And this interaction then leads to a second signal being sent to the interior of the cell. And together, these two signals lead to T cell activation. So a number of other things get triggered when T cells get activated, one of which is secretion of IL-2, which is a very important T cell activation. But the other things that happen downstream of that is that there are actually counter-regulatory mechanisms to control this activation. And so once the T cell gets activated, then there is also the expression of this other molecule called CTLA-4. And that CTLA-4 gets upregulated on the T cell. And now this is binding CD80 or 86, which I told you before was binding this other stimulatory molecule, CD28. And it turns out that CTLA-4 is 20 times more, has 20 times greater affinity for the same molecule. So it's able to outcompete CD28. And by binding this, it is sending an inhibitory signal to the T cell and putting on the brakes. So it turns out this was the first such signal that was discovered, and that was followed by the discovery of another molecule called PD-1, which is expressed on activated T cells and binds either, it's called B7H1 here, but it's actually also called, more commonly called now PD-L1, which is the ligand for PD-1. And this interaction also leads to an inhibitory signal. So that there are various different ways that you can send inhibitory signals or put the brakes on the car that you started. Okay, so what scientists did then was they created these monoclonal antibodies. The first was to CTLA-4, and with CTLA-4 antibody, you could block this interaction. So two negatives make a positive. So you block the negative, the inhibitory interaction, and so then the T cell remains activated. And the same is true for PD-1. This PD-1 antibody blocks this interaction, and likewise, the newer antibodies that bind to PD-L1 are doing the same thing. They're blocking this interaction and releasing the brakes. Okay, so where are we in 2018? There are now a number of FDA-approved immune checkpoint inhibitors. The first, as I told you, was Ipilimumab, or anti-CTLA-4. There are two anti-PD-1 antibodies being used. One is called pembrolizumab, and the other is nivolumab. And then there are three anti-PD-L1 antibodies, adalimumab, avelumab, and durvalumab. So what are these approved for? They're listed on this slide, and you can see that there are a large number of different cancer types that the FDA has approved these various different agents for, and all pretty much in the setting of advanced or metastatic cancer. Interestingly, I just want to point out at the bottom that when anti-PD-1 was approved for treatment of metastatic solid tumors with mismatch repair deficiency or microsatellite instability, this was the first time that a drug was approved for something where it wasn't specifically a cancer in a specific site. So I'm going to take a moment and review one of the ways in which response to cancer therapy is measured, and that's by these criteria called RECIST. And basically, the bottom line is that you need to have measurable disease by some imaging modality. So you can identify the tumor that you're trying to improve, and then you use your agent drug of interest, and you can look at up to 5 target lesions and then classify people as either complete responders, partial responders, people with stable disease, or progressive disease. So I just wanted to review that for you before I showed you this. And so these are from some data from some early trials in different types of cancer using PD-1 or anti-PD-1 or anti-PD-L1 monotherapy. And these are sort of standard spider plots. And what's on the y-axis is the tumor size. So we talked about using measurable disease and the difference between the baseline measurement and the post-treatment measurement. And you can see that some of these lines go up, which means the tumor's increasing in size. Some of the lines go down, which means the tumor's regressing, right? And so if we look at one of these specific examples of lung cancer, and many of you already know that this is the leading cause of cancer mortality in the US and the world today. 80% are non-small cell lung cancers, and the standard treatment is surgery, radiation, chemotherapy, and treatment response has plateaued. For advanced disease, which is how these cancers usually present, there are some other options, including targeted therapy and, of course, immunotherapies, which I've shown you. But I wanted to show you some data from some of these studies. And so the first one, this is a little bit of a complicated slide, but I'll talk you through it. And so the first agent that we're looking at is nivolumab, which is an anti-PD-1 agent. And what I want you to focus your attention on are the second two columns. So the first line gives you the immune checkpoint inhibitor median overall survival compared to the standard of care, which is docetaxel. And second line shows the objective response rate by RECIST, the criteria that we just talked about. So, and then the bottom line is the median response duration, which is not reached for the checkpoint inhibitor, but 8 months for docetaxel. And so if we look at results from other clinical trials, and this is not meant to be comprehensive or anything, just to give you examples, you can see, for instance, similar results where you see an improvement in the median overall survival, improvement in the objective response rate by RECIST, and improvement in the response duration. So you can look at things, either the glass is half full or the glass is half empty. One way to look at this is, isn't it fantastic? We have this improvement in response rates for using checkpoint inhibitors, but you look at this number and it's pretty crummy. And this number is sort of twice as good as crummy. So I think that that's another way to look at it. And given that these therapies are pretty expensive, they range from $100,000 to $150,000 a year to treat a patient with these drugs. This raises the important questions for the field, right? That who will respond? Can we identify those people who are most likely to respond so that we can spend our resources accordingly? And so far, we don't know the answers to any of these questions. We also don't know when to really stop treating. Is there a way to predict relapse and or metastases? These are all great questions, which I don't have any answers to. So now I'm going to switch gears a little bit, and I'm going to tell you about the Cancer Immunotherapy Trials Network. So this is a network that was funded, and I think it's a little bit important to note that it was funded in about 2010, and it's led by Max Cheever, who's the PI, based at the Hutch. And so this was before cancer immunotherapy was, the breakthrough of the year. This was probably, and most of you who know about writing for NIH grants know that there's a long lead time before you actually get the money. And so this was pre-even Ipilimumab being approved. And so the mission of this network was then to design and conduct novel early phase trials to demonstrate proof of concept so that it could lead to pivotal phase 3 trials of promising immunotherapy agents. And so part of this then was MAC needed to establish a network of leading immunotherapists who could design and conduct these trials and conduct trials that were going to generate high-quality biomarker and immunogenicity data. So how did they know what they were going to start looking at? Well, it turns out that immunotherapists have been at this for a pretty long time. And starting in 2007, there were two National Cancer Institute workshops that identified agents that were thought to be the most compelling for patient benefit in the near term. And I'm not going to go through this whole slide, but the ones that are in bold are the ones that were selected by MAC and the executive committee to move forward and to test in the CITN. So how does this relate to anything or to me? Well, it turns out that I was tasked with leading this effort to develop high-quality biomarker and immunogenicity data for the CITN. And in order to do this, and I knew that this was going to be a big job, so I needed some help. And I want to say thank you to Minjin Apodaca, who's still with us and is in the Immunology lab, for agreeing to go with me on this adventure to take on this big challenge. So in 2011, we were tasked with establishing the central lab for this network of immunotherapy trials. And so the charges were to identify and implement a specimen tracking system, establish procedures for specimen preparation and shipment from clinical sites, develop and validate to GCLP level assays to measure immune responsiveness to multiple agents, establish a biorepository, create lab manuals for each study and SOPs for all procedures, and specifically, before enrollment, we needed to prepare lab portions of the clinical protocol, prepare the lab manual for the sites. During enrollment, track all the shipments, provide and replenish all the collection and shipping supplies, process the samples, store the samples in a local repository, generate study data, distribute samples to other labs, provide periodic reports of the study results, publish monthly repository reports, and then during and after enrollment, analyze data, prepare abstracts and manuscripts, among other things. So there was a lot to do. And the initial CITN trials, initial 10 CITN trials are listed here. And mainly the point is just to show you that it was a variety of different types of solid tumors, a number of different agents, some studies which are still ongoing, many of which are complete and are really in the data analysis stage. So as of the summer of 2014, when Minjin and I left our full-time effort doing this and moved to the Immunology lab, the central lab was up and running, and 7 trials had been initiated, approved, and were enrolling. And as of this month, there are 13 active or completed trials. More than 300 patients have been enrolled, more than 50,000 samples stored, more than 1,000 results generated using these assays I described, and new trials are being planned. So it's exciting to see. And I just wanted to take a moment to tell you a little bit about the immune assays that we focused on. They were pretty basic because we were applying them to multiple different types of clinical trials, mostly to measure quantitatively the expansion of different subsets of peripheral blood mononuclear cells and also subsets of T cells, including regulatory T cells and activated T cells, and then also a functional assay, which was the interferon gamma ELISpot assay. So I'm not going to spend much time talking about those, but I will tell you about one of the early trials that we worked on, which was a phase 1 study of subcutaneous recombinant human IL-15 in advanced solid tumors. And the primary objective of the study was to determine the maximum tolerated dose of this agent, and the schema for administration is shown here, where you can see that these were actually daily injections subcutaneously of IL-15 5 days a week for 2 weeks in a row, followed by 2 weeks of rest, and there were a couple of blood draws, 3 blood draws there. And so what is IL-15, you may be asking, for those of you who aren't familiar with it. IL-15 is related to IL-2. It's in the same cytokine family. And you can see by this cartoon that they share receptors for the cytokines. That is, that the IL-2/15 receptor beta chain and the gamma C chains are common to both molecules, but otherwise, in some ways, they're quite different. And so you probably all know about IL-2. It's an important T cell growth factor. It's a soluble cytokine, and it's secreted by activated T cells and binds the high affinity T cell receptor, which includes the IL-2 receptor alpha chain on the surface of these activated T cells. And so this is very important. The other thing I should say is that IL-2 is also important for the activation of NK cells, and I'll talk about that in a little bit. But IL-15 is different in that it's a membrane-bound molecule, and it is expressed by monocytes and dendritic cells, and it's presented on the surface in conjunction with the IL-15 receptor alpha chain. So these two molecules together then are presented to NK cells and T cells that are expressing the beta and the gamma C portions of the receptor. So that's a little bit about how they are similar and different from IL-2. But summarized here is that IL-15 primarily is of interest because it stimulates adaptive immunity, and also NK cells as well, T cells and NK cells. And it induces cytotoxic T lymphocytes, which immunotherapists believe are very important for anti-cancer immunity. It supports the survival of memory CD8 T cells. But it has a couple of important differences from IL-2. And one of them is that it doesn't induce activation-induced cell death, and it's not involved in maintaining these immunosuppressive regulatory T cells. So there were some advantages over IL-2. So when we treated patients with the recombinant human IL-15 from the NCI, some of the results are shown here, and you can see that we're basically looking at NK cells here that are expressing CD56 on the y-axis. And this is kind of a typical profile of flow cytometric profile of untreated patients. But after IL-15 treatment, you can see that there's been this expansion of both CD56 dim and CD56 bright NK cells, which is even more pronounced at day 15. And so summation of this data is shown on this slide, where we're looking basically at the average of patients who were enrolled and treated with various doses of IL-15. And at the highest dose, you can see is shown in blue, the immune response or the NK cell expansion was greatest during cycle 1. And cycle 1, because we know that this is the duration of IL-15 treatment here, and this is cycle 2, the second round of treatment. And if we look at CD8 T cell responses, this is a little bit different because each of these lines indicates the results from an individual patient, either the 2 microgram per kilogram dose or the 3 microgram per kilogram dose in blue. And so you can see that the results were not as impressive as with the NK cells. And that's sort of summarized here, where we show the fold increase from baseline to maximum during cycle 1 of NK cells and CD8 T cells. And so you can see that overall at the 2 highest dose cohorts that the expansion was quite robust, more modest for the CD8 T cells, but still present. And when we look at the subsets of NK cells, you can see that the bright NK cells, which traditionally have been thought to be cytokine-producing NK cells, expanded, as did the cytolytic CD56 dim NK cells. And again, looking at the fold change, what was really impressive was the number of CD56 bright NK cells that were expanded up to 75-fold approximately in the highest dose cohort that was treated. And the CD56 dim ones weren't bad either. They were up to 8 and 10-fold. So that work was recently published in Clinical Cancer Research, and we also conducted another study using an IL-15 agent that was generated by Altor Bioscience. And the easiest way to name this is to call it the IL-15 superagonist. It's way more complicated to talk about this, but I will explain that what this is it's an engineered IL-15 molecule, in that there are actually two IL-15 molecules that have a single amino acid mutation that allows it to have greater affinity for binding to the IL-15 receptor alpha chain. So that a portion of that is then fused to an IgG1 Fc domain. And so all of this then allows the molecule to have a much longer half-life. So it can be dosed once a week instead of every day, as I described for the other trial. And so we tested this in CITN 06, giving it weekly subcutaneously, and we had very similar results to the ones that I showed you for the other trial. Unfortunately, the data are under review, and I've not permitted to show that data to you. But to summarize the work in IL-15, I would say we've tested this recombinant human IL-15, that's naked IL-15 from E. coli, and seen encouraging results. And this is now being tested in a combination trial with both nivolumab and ipilimumab, then that's ongoing. And this Altor product, ALT-803, which we tested in CITN 06, has been combined with nivolumab already, and that's the results were just published this month in Lancet Oncology, and had encouraging results in terms of partial response in about 30% of treated patients. And there's yet another heterodimeric IL-15 that's under investigation in an ongoing clinical trial. And if you search clinicaltrials.gov, there are actually 27 IL-15 trials listed, and so we'll see what some of those additional studies show. So a couple of years ago, when I was talking to Mac and I was giving a different talk, he actually told me this, and he said, in the future, anti-PD-1 or anti-PD-L1 therapy will be considered a core therapy for subsets of every type of cancer, and that the most common category of cancer patients will be on anti-PD-1 or PD-L1, but progressing or relapsing. And I think that that's really been borne out and is true. So really, the next step is combination immunotherapy trials, and searching clinicaltrials.gov, you can find at least 82 of them listed. I think that that's just the tip of the iceberg. I think there are many, many more being planned. Within the CITN, all the future trials are planned with checkpoint inhibitors, and they're being combined with agents that are like IL-15, that are T-cell growth factors, and other immunomodulatory factors. So the questions still remain. Who will respond? Who are the best people to treat? When do we stop treating? And is there a way to predict relapse and/or metastases? So that leads us to the really urgent need for new biomarkers. And I'm going to talk a little bit about each of these. I think we heard a really nice talk from Thomas Long about mutational analyses in the setting of skin cancers a couple months ago. But in terms of tumor biomarkers, I think there's been a lot of interest in looking at CD8 T cell infiltration and really enumerating the numbers of CD8 T cells that have gotten into tumors, and whether that's a sign that those tumors are likely to respond to immunotherapies. There's been even a proposal to replace the traditional TNM staging system with the Immunoscore, which incorporates the CD8 T cell infiltration. I think with the approval, a lot of these checkpoint inhibitors, there was a lot of interest in using PD-L1 expression in the tumor as a way of identifying people who might be the most responsive. So PD-L1, as you remember, is the ligand for PD-1, and upregulation of that might be suggestive that there's something immunological going on in the tumor, and therefore those tumors might be more amenable to immunotherapies. But it turns out that people who don't really have PD-L1 expression can still respond to checkpoint inhibitors. So while there's this correspondence between PD-L1 expression, higher PD-L1 expression you're more likely to see a response to checkpoint inhibition or immunotherapies, but you also can't exclude people on the basis of this marker. And then the mutational analyses, I think, are an area of really great interest. Even just this month, there was a paper in Cancer Cell describing analyses from a prospective study of patients who are getting treated with a combination of Ipilimumab and Nivolumab, and showing that those who had higher mutational burden were more likely to respond to that therapy, combination therapy. And then there's the issue of the microbiome. I think that everybody is aware that the gut microbiome is super important. And it turns out that it is also important for cancer immunotherapy, and that there are profiles that are more consistent with those people who are going to respond. And it's not just the gut microbiome. It's actually, people are starting to publish papers looking at what we traditionally would consider to be sterile sites that now have been discovered to have communities of bacteria associated that either predispose or detract from successful cancer immunotherapy. And those studies are primarily in animal models, but there's definitely data in human. And I think that this is an area of great interest and also great complexity. And then finally, I think all of us would like to see biomarkers found in the peripheral blood. That's kind of our business of testing things in the peripheral blood. And one of the markers that I'm particularly interested in is in myeloid-derived suppressor cells. And so what are these? This is a heterogeneous group of immature myeloid cells. And they're important in normal immune homeostasis. They limit inflammation normally. We all have them, not very many of them, but we all have them. And interestingly, there are more of them in women who are pregnant. And if you recall, a fetus is an allograft, and it's not self. So there needs to be a means of maintaining that pregnancy. And myeloid-derived suppressor cells seem to be important in that. They're highly immunosuppressive for T cells and NK cells and inhibit their proliferation. They're found in the tumor microenvironment. In fact, the tumor microenvironment, which is hypoxic and has chemokines and growth factors that MDSCs seem to like, attract them there. And so they have a role in tumor progression and metastasis, and also have been described to be important in other important general areas like autoimmunity, infection, and transplantation. So if you search PubMed, for the last couple years, there have been over 500 studies. If you search for MDSCs, you'll find over 500 articles each year for that. And I think that's just going to continue. So just to give you a little bit of a flavor of what they're like, it turns out that there are two flavors of these MDSCs. There are ones that are more granulocytic and ones that are more monocytic. But regardless of that, if you look in pretty much every type of cancer, you can find these cells circulating. And these levels of these cells seem to increase with age, and they negatively correlate with things like tumor stage, metastatic burden, response to therapy, and progression-free survival or overall survival. So if you look at some data from some of these papers, this is one of them where investigators looked at metastatic melanoma and clinical outcome in terms of overall survival. And you can see that that dark black line are the patients who had metastatic disease but had low levels of MDSCs, and they had better survival. When they looked at regulatory T cells, there was no effect on survival. And similar data are shown here in the setting of melanoma and Ipilimumab treatment, whether you tested people before treatment or during and after treatment. Low monocytic myeloid-derived suppressor cell levels predicted better overall survival. So we in the Immunology lab have now developed a whole blood assay for these cells. It's a bit complicated. There are a lot of markers. Mostly we have used markers for what it's not. and what it is. And I'm not going to go into the details really of this, but we feel that it's a quite robust assay, and hope to get our data on the preclinical variables that affect quantitation of this important cell subset submitted soon. So my conclusion for this part of the talk is that I think that myeloid-derived suppressor cells are very promising, and the early results are promising for prognostication. There are more studies needed for utility. There's a lot of interest in this area. If you search clinicaltrials.gov, there's about 60 trials that are either looking at MDSCs as a biomarker or targeting them for depletion in order to try to improve the outcome of immunotherapy. So to conclude, I would say that we're in a new age for cancer therapy. Immune checkpoint inhibitors are here to stay for sure. Of course, CAR T cells, as I mentioned before, and really the name of the game is combinations of these agents. What are going to be the best ways to get the best results with the least toxicity? And I think it's very clear that laboratory medicine can play a very important role in developing clinical biomarkers for monitoring and prognostication. So, and from our lab's perspective, we're very interested in developing more biomarkers that may be important that use flow cytometry as a way to measure, not just quantify different cell subsets, but also to measure their function. So I'd like to just acknowledge the support that I've gotten from Minjin, Kathy, and Mark in the clinical immunology lab. They've been incredibly supportive as I've been involved in these CITN efforts. And then from the CITN, Mac Cheever, the PI, and Steve Fling, who took over for me as the leader of the lab there, members of the CITN Central Lab and the coordinating center, and then also acknowledge the role that Steve DeRosa, who's a faculty member in our department, on how he and John Harrell at the HIV Vaccine Trials Network helped us when we set up the CITN Central Lab.
My last remaining question that you didn't answer is, can you say a little bit about autoimmune complications of checkpoint inhibitors? Sounds like they're very therapeutically effective, but what's the downside been?
For sure, there are various different types of autoimmune complications. I mean, I can't really give you the numbers, but I think that pretty much any organ system that you want to name, I think pneumonitis is a big one, but I think that that is unfortunately the downside of all of these therapies, is that you can induce autoimmunity. I'm sorry, I'm not more specific than that, but.
Yeah, so on your measuring your level of the peripheral blood of your MDSCs, does that actually correlate then with what might be in the tumor? So is that going to be a significant marker if you're looking at how these cells might actually have a role?
Yeah, I mean, I think that that's a really tricky. I don't know that there's been a good correlation made between tumor MDSCs and peripheral blood MDSCs. I would say most of the studies are really in the periphery because obviously it's easy to access blood. The other thing that makes it complicated is just the number of markers that you need to identify these cells, right, which you can do using multi-parameter flow. But if you want to do that in the tumor, then you have to actually digest the tumor. You can't use any of those markers because a lot of those markers overlap with other cell types that are going to be in the tumor too, potentially. So without the advent of sort of multispectral IHC, which I know there are definitely people who are working on that, then there might be better studies eventually looking at those two things together and correlating them. I think, so does that kind of answer your question? It's a difficult issue.
Thanks, Chihiro. A philosophical question. I feel like antibodies are big, and they kind of have a hard time getting into a tumor space. Are we making any progress on more small molecule kinds of checkpoint inhibitors? Or are we always going to have to rely on these bigger antibody-based things?
I'm trying to understand your question. If there's a way to maybe increase, use smaller molecules so that there's better access
Right.
for having the wanted effects on.
And they would be less expensive, right? I assume.
That's a question for pharma that I think that I couldn't answer. I don't think that the cost of the agent has anything to do with how hard it is to make the agent. But no, I don't know of any efforts in that regard. Sorry.
Thank you very much for the talk. I was kind of wondering how NK cells compare to CD8s at killing cancer cells, because like CD8s are really good at it if they can get to the cancer cell.
And I would say that NK cells can be too. Of course, we know that it depends on the expression of class 1, right? So, so I think it's going to vary. But I think the other thing that I didn't bring up that I should have, I think, is I was talking all about CD8 T cells, and then the IL-15's main, effect was on NK cells, and that was, I would say, not totally expected. I would say the preclinical models predicted that there would be more effects on the CD8 T cells, so we were a little bit surprised at that. I think, but I think in terms of, how it works. I think that there's been a lot of discussion about whether it would be best to combine it with antibodies to induce ADCC, because and if you think about how NK cells work, so I think that to answer your question, are NK cells better than CD8 T cells? I think. I can't really answer that question, but I would say that I think the more that you can get of both, then the better. It's sort of like whether or not you think that it would be useful to have like a vaccine against a tumor antigen, where you're stimulating a response to like a single protein or single peptide. And I would say that based on what I know, that I think everybody would rely upon that response being so strong that it spreads, and there's epitope spreading, so you're getting a polyclonal response, right, to multiple antigens, and that's how you're really going to see an effect. And so, in the same vein, right, NK cells are killing, can kill tumors, and CD8 T cells can kill tumors, and I think you really want. The best possible thing is to induce both.
Can I have a follow-up question on that? So you said the PBMCs, right? They're very accessible. They're good for lab medicine, but there's a lot of interest in resident memory T cells. And I was wondering, if you digest a tumor, does the PBMC picture, this NK predominant response, where the NK cells were better than the CD8s, if you look in the tissue, are the CD8s in fact doing more because they've gone to the tumor, but they're stuck there and they no longer circulate?
Okay, so that's a really good question about, right, we're testing things in the peripheral blood, and because we can, and we really care about what's happening is in the tumor. And obviously in humans, that's really hard to do. So I think that some of the studies, though, that have been done by Tom Waldman, where he did sort of kinetic studies, show that, if I'm trying to address your question, is that that there is evidence to suggest that the T cells are actually going to the tumor, and that's why we don't see so much of them in the peripheral blood. And so, we've also looked at functional markers of proliferation in the peripheral blood, and you can see that the NK cells are proliferating and the CD8 T cells are proliferating. by doing like Ki-67 staining. And so what that suggests is that the amount of CD8 T cells that you're quantifying in the peripheral blood is actually an underestimate of what your effect is. But you need to do obviously more studies looking at homing markers and things like that to be able to say, and then look in the tumor and show that they're actually there. And we certainly haven't done those studies, and I think you have to get tumor tissue to do that, which I think that it's we've tried to do some studies where we're trying to get tumors. That's why I think melanoma is a very popular disease to study, because it's easy to get the tumor. These other ones, it's really hard to get people interested in participating. So.
Great talk, Chihiro. So I'm going to ask a genomics mutation question, right? So my question is, for the CITN lab, this core lab, are there any assays around MSI, total mutation burden, any other kind of genomics, or is that being done by another lab and then compared to the immune subset analyses that are being done?
Right. So I think that there are collaborations being set up, and I think the name of the game, honestly, is who's willing to do this for free. Because the NCI does not give the CITN a lot of money to do this. And so I actually know that there's some gene expression collaborations that have been undertaken. But the other thing that I should say is that although we don't have very much tumor tissue, there are a few studies that collected a little bit of tumor tissue. If there was interest in looking at non-tumor tissue, then there basically is a very large repository of peripheral blood mononuclear cells where investigators who are interested can certainly come to us and ask for access to those samples and get those samples to test. But it unfortunately at this point in time doesn't come with funding from the CITN or anything like that.
That's too bad, yeah, because it's really interesting. It was fantastic. I learned so much about the immune subset and those kinds of analyses, and we already have biomarkers we're using in clinical practice. MSI, as you so clearly stated, the first tumor agnostic biomarker, which is huge, and that's just for our, through the genetics division we offer MSI testing, and that's just skyrocketed our volume, not surprisingly, because now MSI is a predictive biomarker for immunotherapy. So, and we've now rapidly put on total mutation burden in our bioinformatics pipeline as an output for Oncoplex because of this. So we're doing this in practice now. It's too bad there's not any funding for it for the trials. It is expensive to do, but it seems like there's a natural marriage between what's happening on the tumor side and what's happening with the immune side that we could think about ways to marry those up for the most predictive suite of biomarkers, so to speak.
Yeah, and I definitely think that Mac and others at the CITN would be very interested in any kind of collaborations to try to advance what we can learn from these samples. I mean, the
really precious resource.
Let's thank Dr. Morishima one more time.